Hi, I'm Tyler DePrano, and I'm going to be doing a microbiology video on the types of viral replication. So, before we start understanding the types of viral replication, we need to know what a virus is. It is a double-stranded or single-stranded RNA or DNA genome that is surrounded by a protein coating called a capsid. In some viruses, the capsid is surrounded by a lipid bilayer that contains viral proteins. This extra layer is called the viral envelope. Now, how do viruses replicate? They need to enter a host cell, they need to attach to the cell membrane, and they need to penetrate it and inject their RNA or DNA genome. This genome is then replicated, and viral proteins are made from it. And then these proteins are all assembled, the viral particles are assembled, and the viruses or the virions are assembled. A virion is a complete virus of the capsid and genome, and these virions are then lysed from the cell or released, and they go on to infect new cells. So how do we classify viruses? We can classify them based on their types of replication. So this is the Baltimore classification system. It was developed by David Baltimore. He won the Nobel Prize, and it classifies viruses depending on the type of genome and how they replicate. So there's group 1 through 7. Now I'm not sure about many of you, and no offense to Dr. Blair, but I was just a little confused on how this was explained in class, so I thought I would make this video, and hopefully this does a little bit better of a job explaining it than the picture that we had. I just wanted to go a little more in depth because it was really confusing when we looked at this. So let's start with group 1, double-stranded DNA viruses. These include the adenoviruses, herpes viruses, and pox viruses. The adenoviruses and herpes viruses replicate. They just enter the nucleus and then replicate just like the host cell does. Their DNA is integrated, converted to mRNA, and then proteins are made from that. Pox viruses come with their own enzymes in the capsid, and those enzymes make the pox genome into mRNA. So it just replicates in the cytoplasm, not in the nucleus. That's the big difference between the pox virus and the other two. Group 2 is positive sense single-stranded DNA viruses, including parvoviridae. These just form, the positive sense goes to double-stranded DNA, and from there it's the same. The double-stranded DNA is converted to mRNA, and then proteins are made from it. This group also includes anellovirus, which is found in nearly all humans, but luckily for us it is asymptomatic, so we are not harmed by it. Group 3 includes double-stranded RNA viruses. Reoviridae fall under this. These are, since they're already double-stranded RNA, that means they include positive sense and negative sense RNA. So mRNA is positive sense RNA. So let's go back to that classification system. mRNA is what all these viruses have to be. They have to convert their genomes to. So mRNA is positive sense RNA. Alright? Now since this group 3 has positive and negative on it, since it's double-stranded, it has a positive sense and a negative sense, so it just gets its positive sense and gets mRNA from that. And then that goes on to be converted into proteins. Group 4 is a bit different. So group 4 is straight up positive sense, and it's single-stranded, and it includes togaviridae, picornaviridae, and flaviviridae. It just enters the host cell and can immediately start being transcribed because positive sense RNA is essentially mRNA. So the proteins are just made immediately. Group 5 is a bit different because it's negative sense single-stranded RNA viruses. So these negative sense RNA viruses need to be converted first into positive sense RNA. So they come with their own enzymes, and these enzymes, because the host cell doesn't have the enzymes necessary to convert negative to positive. So they come with their own enzymes, and these convert the negative sense into positive, which is then going to be translated into proteins. So these negative sense include things like influenza, flu, and rabies. The only real advantage that these have is that they can make a lot. So down here you see the negative goes to positive. Well, then this positive can be made into proteins, or if we come down here, it can be made into negative RNA again and go back and start this whole process over. But I would definitely say that group 4 is far more advantageous in the fact that it can just enter the cell and start being translated immediately. Group 6 includes positive sense single-stranded RNA RT, which is reverse transcriptase viruses. So these include retroviridae, which fall under HIV. So if you can see this chart here, they start off as RNA, and the RNA uses reverse transcriptase that comes within its capsid, so it brings this enzyme into the cell with it and converts it into negative stranded DNA that's made double-stranded. This DNA then goes on integrate into the host and replicate normally as mRNA and then into protein. Group 7 is also going to be different. So it's double-stranded DNA, RT again, so reverse transcriptase viruses. These include HEPA DNA viridae. So one of the viruses you'd find in this category or this family is hepatitis B. So this one's also strange. Since it's double-stranded DNA, it could just go straight into the host, but what you need to know is that this DNA has a gap in it. So it's a bit different. So this DNA will first be converted into RNA. This RNA will use reverse transcriptase to become DNA, and then it becomes integrated in the host and is transcribed into mRNA and then translated into protein. So that's how hepatitis B would replicate. We don't know why it does this. It just worked and still works. And that's how it is. So I hope that this video helped and that broke it down a little bit better. I know it's not the best, but hopefully this can help some future students. Dr. Blair, if you need to use this for your students, you are welcome to. Thank you.